This is a tutorial video for Cyrus Bench Homology, hm.cyrusbio.com, for how to use that tool to generate a model of a symmetric homodimer protein with a C2 symmetry. This is a really common type of symmetry in the world of protein structure. So let's go ahead and imagine we have an input sequence that we've gotten. Don't know anything about it yet. I have the protein sequence. So I'm going to blast P. So you can get to this just by Googling protein blast, for example, and then click that first link. And here I am at blast P. And I've gotten the sequence that I'm interested in. And probably in real life I would already know something about this sequence, but let's say I don't know anything about it yet. So I'm just going to paste that sequence in there that I copied, for example, from a FASTA file. And I'm interested in structures, so instead of searching the whole database of NR, I am going to search the PDB, the protein data bank, those structures that have, those sequences that have structures, and I'm going to go ahead and run that. And that takes a moment to run, so I've already run that here in another window. And here's what comes out, and let's see. So we can see specific hits. This is this PPAR. Let's say I don't really know what that is yet. But that seems to be really an important family that I've hit. And wow, that's a lot of hits. This must be an important target. And here we go. So the first one, chain A, crystal structure of the ligand binding domain of human PPAR gamma in complex with an agonist. And that has this accession number. And oh wow, there's really a lot of these. And so in real life, you probably already know something about this. And so you may know the PPAR family. This is actually a family that's relevant in for drugs in diabetes, and there's some drugs that target PPAR. In this particular example, I didn't say it yet, but this particular example that I put in is actually the hamster PPAR sequence. You notice here there's a lot from other organisms, most of them human, which makes sense, right? So now let's say I want to use that PPAR, and I actually happen to know for PPAR from my pharmacological needs that I'm looking for something that has a sequence that's relevant as a gamma alpha dual agonist, because I've seen in the literature that that's particularly interesting as a pharmacological target. So let's go in the PDB and find a nice template to go off of, and I'm going to do PPAR dual agonist, based again on a little bit of knowledge that I have. And then, you know, you could go through these and look at the structures, and here I've already reviewed the literature a little bit, and I may have discovered that I'm interested in the version that came out of this paper because the complexes here are relevant to my pharmacological needs. So now I've decided that 2Q8S is what I'm going to template off of. Let's take a quick look at it here. It's cyclic. It has a C2 symmetry. So I can go ahead and download that PDB, and I'm going to save that for later. So here we go. Let's download that PDB, and let's download the FASTA sequence. And now what I want to do is I basically want to make a symmetric model that has a C2 symmetry that templates off of the oligomeric state of this 2Q8S. So I've downloaded that sequence, and then I can copy paste that and put that in here. So here I am. We're going to run the monomer in Cyrus Bench. So the next step now is I'm actually just going to paste that sequence in. I've already done that here. Put that in and run. Here there's a little bit of a hiccup on the back end. Took a few days to run, but it comes out. So now I'm running as a monomer, right? So I've identified 2Q8S as a sequence similar to my sequence, and I'm putting in my sequence, which is the hamster PPAR sequence. And then I can look at these structures, and let's click on one of them. So you've seen this before. This is what the structures look like, but of course this isn't symmetrized yet. In general, as you've learned before, the rank 1 model is the best model. So I'm just going to go ahead and download all models. Now I have a monomer model, and now I need to symmetrize that. So I'm going to open up the top structure, and I'm going to go ahead. I've downloaded that. I'm going to open up model 1 in a molecular viewer, one like Pymol, for example, and then I'm going to superimpose. I'm going to make 2 copies of this particular monomer, and then I'm going to superimpose one on chain A and superimpose one on chain B. So let's go ahead. I'm going to open that up that I've already done here. So let's open up this guy. So I have that in a PyMol session. Let's move that in here. So all I did is I copied the model 1 file, and then in here, in my file editor, in my operating system, I've made a copy, model 1 and model 1 copy. And now I can open up 2Q8S, which I'm templating on, and then I can superimpose model 1 onto chain A of 2Q8S. So that would be a command like this one. Align model 1 to 2Q8S with 0 cycles. And then I'm going to do the same thing for model 1 copy onto chain B, like this. So I've already done this before. Let's run it again. And there you go. And so now here in light blue is 2Q8S. So that's the actual human structure. And then now I have my models, right? So I can turn this off. This would be chain A. This would be chain B. This is already pretty good. This is not more than pretty good. This is already pretty awesome result. So I already have a pseudo-symmetrized, close to symmetrized version of this structure. But I might have some weird aspects, especially this interface, because I've just naively superimposed these. So now I'm going to go ahead and I'm going to save these molecules. I'm going to save model 1 and model 1 copy as the symmetrized. I'm going to save a separate copy of each one. I've already done that here, so let's go back over to my operating system, and here in another directory I've saved those, model 1 copy symmetrized, model 1 sim, model 1 symmetrized, and now I can go ahead and open those. Now I have those, and I can combine them into, just by copy paste, into a final structure. So I just, this one here, oops, that's not what I wanted to do. This one here, I am gonna, sorry about that, so I'm gonna open that up in a viewer. So model 1 copy, I've just gone through and I've replaced chain A with B. This is just find and replace in a text editor. And then I've gone ahead and pasted together, made one master one, where I have model 1, and then I have I just put one line. So I just had model 1, and then I put one line ter, and then I paste in this copy, which is now chain B, paste that in. This is literally just copy paste. And now I save that, and I've saved that as output sim. So now this is one PDB file that contains my initial symmetrized model of these backbones without having optimized the side chains yet. Now for the next step, to fix the side chains, I'm going to open up CAD here. Now I just need to fix the side chains in a fixed backbone context. So here I have all my projects, and let's sort these. So I'm going to do some cleanups. I've already done this before of 2Q8S. And here you go. So I just uploaded that structure from that output sim directly from my hard drive. And if you remember, that would just be a new structure and then load from file, right? And then I would get the file that I want. So I've already loaded that. That's already scored. And then all I did is I take that and I do a repack to fix the side chains. I did 5 repacks, and then I'm just going to take the lowest structure, happens to be energy structure, structure 3, and then I just click that one, download the selected PDB, and let's go back to the Finder. So there you go. So that's this output, that's this final output sim repack, and then that's my final model where I fixed the side chains and left the backbone intact in this very close pseudo-symmetric state. So that's the final output. Let's open that up. Oops. Let's quit PyMol here. Let's open that up. And there you go. So there I have that overlaid onto 2Q8S. I can color it, for example, by chain. Go ahead and do that. And there you see it overlaid onto, let's probably color 2Q8S, dark blue, for example. And there you go. So now I have a model that's templated off of 2Q8S. You also notice now I've left the backbone intact, but I've fixed side chains at the interface here, so they make more sense in terms of the model. And one of the cool things is we've actually built out ab initio, built out some loop that didn't appear here into Q8S. This is a high quality symmetric model of a homodimer templated on a close homolog from another species. And there are a lot of C2 dimers out there in the universe, and this is how you can build models for them. Thanks for watching.